The drug development process that we know is failing us dramatically. The statistics are pretty nuts. 1 in 10,000 compounds makes it to an FDA-approved drug with an average cost of $2 billion, taking on average 15 years.
Drug development has a 90% failure rate. It's expensive, it's slow, and it still relies on animal testing developed decades ago. So today we're talking about the new science that could change all of that. Kayla Ellsworth is the Scientific Operations Coordinator at Speccro, a biotech consulting startup using new approach methodologies, otherwise known as NAMs, to help drug developers get better answers faster and at a fraction of the cost. She's a neuroscience grad who networked her way into one of biotech's most exciting startups, and she's just getting started. And later, Kayla is going to share what it's like walking into rooms that are 95% male, and about the moment that completely changed how she sees her place in this industry. Please welcome Kayla Ellsworth, Scientific Operations Coordinator at Speccro. Let's get into it. Hi Kayla, how are you? Thank you for joining me for this episode. I'm looking forward to hearing all about NAMs. No one knows what that means, but they're going to find out. So welcome.
Thank you so much for having me, Heath. How are you?
I'm really good. I want to give you this opportunity to first introduce yourself and your role at Speccro and talk a little bit about Speccro and give people a little insight as to where you're coming from.
Yeah, absolutely. I can give you my career origin story if you'd like.
Let's hear it.
Perfect. I studied neuroscience in university, and I knew I wanted to break into the biotech industry, but the question really was how, because it just happened to be the most capital scarce time in recent history. And on top of that, on top of that, I was also seeing all my friends, apply on LinkedIn for months and months and months and just not seeing a lot of success there. So I knew I had to do something a little different. And by a stroke of luck, my home base is in Boston, which just happens to be the center of the universe for anything biotech related.
Yep, absolutely.
So I hit gold there accidentally. And I plugged into ChatGPT. Okay, what life science conferences are happening over the next few months in Boston? And I reached out to the conference coordinators and basically said, hi, I am a broke post-university student who's just looking to learn and meet people. Will you please take pity on me and give me a free pass? Because these conferences are thousands and thousands of dollars.
Yeah.
And to my surprise, they said yes. So really shout out to those coordinators for opening the door to where I am today. And that first conference, I kind of had that feeling when you get transferred to a new school. I was like, everyone's looking at me. They know I don't belong here.
Fish out of water.
Exactly. It was definitely fake it till you make it a little
syndrome,
bit. Yeah.
All of that.
So I went up to basically everyone I could, introduced myself, and really just networked my little heart out. And that's where I met Shashi Ramaya, who is the founder and CEO of Speccro, who you just interviewed. And he basically just said he was willing to take a chance on me. And not only is he my employer, but he has been such a mentor to me. He's really shaped my career thus far in the industry.
Amazing. What a great opportunity.
True.
And that's just like, well, that's just it. It's just putting yourself in a kind of a weird, uncomfortable place,
Totally.
and where you can meet other people who have sort of similar interests and ideas, and that's, look where it got you.
Absolutely.
Right? Had you not taken that step, right?
Yeah, I knew absolutely zilch nothing about drug development and what goes into it, and NAMs, which we're going to get into later. But I feel like it just taking that risk and putting myself in those uncomfortable positions just really led me to such a fruitful experience and, a lot of learning and opportunity.
So tell us about Speccro, and what was it about that company and what Shashi is working on that was so compelling and interesting to you?
So when I met Shashi, he introduced this concept of NAMs to me, which are new approach methodologies. And like I said, I had no idea what these were. And what really drew me to them initially was that they shift the emphasis away from testing on animals, which is the traditional model.
Right.
And, I love animals and I love science. And, historically, those go hand in hand.
Right.
And a lot of animals have, unfortunately, suffered and died for the sake of human medicine. And it seems to have been a necessary evil, but it's 2026 now, and there should be better ways, and there are better ways. And I really wanted to be a part of that sort of boat that's rowing everyone forward in this direction. At Speccro, we are a consulting operation in preclinical development. And Shashi has curated a team of industry-leading experts across the different sectors of drug development, whether that be ADME, PK, safety, toxicology, regulatory, to answer questions and attack problems at truly every angle. And we help companies do this by incorporating these new approach methodologies, NAMs, into their drug development process to answer those questions, solve those problems in a more predictive, accurate, timely, and cost-effective way.
Well, it's not unusual for, particularly in the healthcare industry, to have done things a certain way for a long period of time, mostly because we didn't really have any alternative options, right? But sometimes we get complacent and we just like, well, that's how we always done it. And animal testing has just been one of those things that's how we've done it. That's how we've always done it. Right. So what else can we do? And so what else can we do? What can we replace that with?
Yeah, absolutely. I would love to get into that. I guess I should probably start by defining what a NAM is.
Yeah.
Now, there are quite a few different definitions here, and this is an evolving field, so the semantics are, flexible and changing. But I would to simplify it, they are models, technologies, and methods that are used to study biological and chemical processes and generate data without relying on traditional animal models. And the idea is that they are faster, cheaper, more predictive for humans, meaning in theory they could better recapitulate human physiology. And that's not to say that animal testing and animal models are not necessary in today's, current structure, but there is still room for improvement, especially when we see the current challenges that we do and the gaps that still occur in drug development.
I mean, how bad is it? Like, what are the stats around it?
Yeah.
That's what everyone's going, well, how bad could this be? Like, what's going on out there?
Well, the statistics are pretty nuts. 1 in 10,000 compounds makes it to an FDA-approved drug with an average cost of $2 billion, taking over 15 years. These issues that we're seeing are really big, hairy problems, and there's not just one thing we can do to solve them because there's so many different factors at play. But, full stop, we need better ways to help patients. And that's where NAMs come into play.
I mean, those are that's what they're all for. I mean, drugs are for patients to resolve problems, save lives, et cetera, et cetera. If there is a better, more efficient, less costly way, we need to find them.
Exactly.
Because we've been doing it this way for a very long time, and that's not a very good success rate.
Exactly.
1 in 10,000. It's terrible. Okay, so how are we going to fix this?
Yeah.
Kayla?
Yeah, well, I'll give you a couple disclaimers. So, first of all, I am not a PhD in this area. I am not scientifically trained in these technologies, but in some ways, I think that can make it easier for me to communicate to a broader audience in a way that, is maybe more digestible. And that's also a non-liability statement as well for the NAMS experts not to come for me if the semantics aren't perfect. But my second disclaimer is that the new in new approach methodologies can be a little misleading because some of these technologies have been around for quite a few years, and some are even validated, meaning that they are proven to be reliable for their fit for purpose use. So those are kind of my two little pre-statements.
That makes sense.
Before I dive into the different flavors of NAMs. And
okay,
there are going to be some things that are considered under the NAM umbrella that I probably won't touch on, but I'll go over some broad categories. I'll start with the ones that I am least familiar with, and then finish with the ones that are more in my wheelhouse.
Sure, sounds good.
Awesome, let's do it. So first we have in silico methods. This encapsulates computational modeling and simulations, which means using computers to analyze, simulate, and predict biological or chemical behavior without running physical experiments. So it's kind of like a forecast as to what might happen in the human body. So for example, you could use in silico to predict whether a compound is likely to be toxic based on its chemical structure. If that all makes sense.
You're able to do that due to the decades of scientific experimentation and data that already exists.
Yes.
The repetitive nature of science is that you often do a lot of the same research leading up to a certain point where it might branch off or right? But
exactly.
This data is something that you can use, and so you're not necessarily going back to ground zero again. You're actually picking up where everyone else has left off.
Right. And okay. I mean, that's really where AI comes into play here, is using, conglomerating all the data that we have to see patterns and use those patterns in a predictive way.
Right.
So the next category that we have is in chemico. This has actually been around for quite a while. There's no living systems involved here. It's just chemical reactions. So This is measuring things like binding, reactivity, stability, all that fun jazz. And next we would have ex vivo. So this is using actual tissues taken from a donor, whether it's human or animal. So for example, you could use human skin samples for irritation testing, or liver slices to study metabolism, things like that.
So that's where you don't have to use mice and rats and other things in order to do those testings, because that's often like medications or ointments or topicals. That's what they have to find out, is how will this impact human skin.
Yeah.
Interesting. So they can actually use human skin, but it's not on a person. They're using
post-mortem, hopefully.
Yeah, interesting.
Yeah. I won't be signing up for that study personally.
No, me either.
Next, you would have omics as a category. So these are technologies that measure large sets of molecules at once. So say you are trying to understand a city. Instead of checking street by street, omics looks at a satellite map of the entire city to see things like traffic patterns, busy areas, what different parts connect. That's what omics does for biology. Broadly, it lets you compare patterns and gives you more of a system-wide insight into how biology is responding. And there are some, it's very interesting, and it's a rapidly growing field right now. And there's different subtypes that sort of fall under this umbrella of omics. I won't go too in depth to those. I'll just mention them briefly. There's genomics, which looks at DNA. There's transcriptomics, which looks at which genes are turned on and off. There's proteomics, looks at which proteins are made and, if they change, how they change. And there's metabolomics, which looks at small molecules called metabolites that represent a sort of snapshot of biological activity.
Very interesting. You're very good at explaining it in a layman's term. I'm serious. Like, it's very good you really create a very good visual of how it works. Thank you.
I appreciate that. And that's like, we can talk about that later, but that's something I'm also passionate about because, sort of being the new kid on the block in this industry, people will often speak to a crowd as if everyone is an expert in the field. And, in some cases that is necessary to sort of skip the small talk in a way. But for someone like me, I want to understand the science. I want to understand what people are saying, but there's sort of a barrier of accessibility there when it comes to the language. So I think, sort of when you're on a podcast, especially like this, you never know who's listening. You're going to have a very broad audience.
You don't know. No.
So I think, going about the verbiage in a way that really is accessible to as many people as possible is, something I'm passionate about.
The science people, the scientists love the science, right? And they can't get enough of it, right?
Yeah.
Whereas, the rest of us are our eyes are glazing over like, wow, it's fascinating, but I can't even comprehend half of it. But it's just, it's a very interesting industry because you have so many different players involved and they're all looking for a different part of the story. The scientists want to hear the science and the investors want to hear about the business, and everyone else wants to hear about the patient outcome. And so like you have all these very interesting perspectives, but and a variety of conversations that are always going on. But it's knowing what to say when you're in the right audience, right?
Exactly.
That's what's important.
Exactly. Yeah. And your podcast does a great job of that too. And, you have such a strength in taking information and then synthesizing it in a very digestible way.
Yeah, well, thanks for saying that because that's what I try to do. I'm not, there's no such thing as a dumb question. We all hear that, but I like asking a lot of dumb questions because often when people are talking about something that they're very familiar with and very passionate about, and it's just something they talk about all day long, they'll shorten things and use acronyms and what have you. And it's just like, okay, I don't know what that acronym is, so you're going to have to explain that. So it's just that reminder. It's like, oh yeah, you don't live in my world or you don't live in my head. So I have to stop.
Totally.
Reassess and then re-explain that. And that's also too where coming in when you're, presenting a pitch for a fundraiser, like you don't spill your science jelly beans all over the lobby when you walk in because everyone's just going to walk on them. And, you gotta hold some back and it's more of a tease. And it's like, here's enough information to generate interest where they want to come back for more without
overwhelming them too.
Mansplaining it.
Yeah. Very, very well put. It really is the acronyms that, have always gotten me and continue to get me. There's a ton of research saying that, in scientific papers, even peer editors, the more acronyms you use, the more confusing it is. It's actually just, even though it's more of a mouthful to say the actual, what it stands for, it sort of clicks in people's heads more.
Yeah, it's important. Yeah, very much so.
Well, I'll sort of get into that last category under the NAMS umbrella, which is more my wheelhouse, which is, I'm going to sort of rephrase that. This would be complex in vitro models. So This is an umbrella term, and there are a lot of different things that fall under this. So I will sort of start from the most basic and go to the most complex. So first we have 2D cell cultures, meaning growing cells in a flat layer, typically at the bottom of a petri dish. And these offer a lot of control over different conditions and variables and provide scale. Now, this is not new. This has been around for decades and decades and decades.
Okay.
Now, more evolved are 3D models. These would include spheroids and organoids, and we'll get into those. But I think it's important to say that real tissues in the human body are 3D, with cells stacked on top of each other, interacting in all different directions. So these models already represent a more realistic architecture.
Interesting.
Yeah. So I can break this down a little bit for you.
Okay. Let's do it.
Let's do it. So spheroids, these are more basic than organoids. So think of a ball-like structure that is a cluster of cells. And there's typically one predominant cell type. For example, it could be breast cancer cells, cardiomyocytes, which are heart muscle cells. Mm-hmm. And these spheroids are widely used in cancer research to study tumor growth, drug penetration, and resistance. Next, which is a little bit more complex, are these organoids. So in these, you're seeing multiple cell types arise, which self-organize into structures that resemble aspects of real organs or organ-specific regions. So I'll give you an example. A gut organoid could mimic the intestinal lining barrier and really measure pathogen entry, absorption, permeability, but it's not representing the whole digestive system. It's just a piece of it. So overall, these 3D models show some functional and structural features of actual tissues in a more static environment.
Okay, so now explain how you use that. How do you apply that?
Well, you can use them for a variety of different things. You could use them for basically anything under the sun. Like I mentioned, spheroids are widely used in tumor research, cancer research. The example I gave of the mini gut, you can see what compounds cross that barrier, sort of are more permeable, and that can give you insights into whether your drug is harmful, whether it's helpful, what those biomarkers are going to look like.
I see. Okay. Interesting.
Things like that. Does that make sense?
Again, you're recreating the part of the body or the area of the body or the cellular, well, being at a cellular level that you would use. You apply it in animal testing.
Exactly.
You're replicating it, but you're actually replicating it in a more human form.
Exactly. So the point of them is to better recapitulate what is really happening in the human body.
Right.
And especially, like I mentioned, these 3D structures represent more realistic interpretations of what's going to happen in the human body.
Right.
Versus just, 2D cell lines.
Right, exactly.
Mm-hmm.
Very cool.
Yeah.
Wow.
And
Very
sort of
cool.
the next rung up in complexity is microphysiological systems. These are also referred to as organ on a chip. There's some arguments and semantics there. I'm going to call them microphysiological systems. You might also hear MPS for short. Now, this is more cutting edge, I guess you could say. Whereas organoids, which I just mentioned, they grow by self-organization. You basically give the cells the right signals and they sort of figure out how to form a mini tissue. Microphysiological systems, on the other hand, are devices that are engineered from the ground up. So microfluidic channels, membranes, and compartments are designed first, and then cells are then placed into specific locations where they then organize and interact under a highly controlled environment.
Wow. That was a mouthful.
Yeah. Yeah.
But I think I got it.
Awesome. And I can
I
can go over some of these key features in more detail. So an important factor of these that differentiates them is that they incorporate flow. So this mimics aspects of, blood flow or other bodily fluids, which already makes it a more dynamic environment compared to more static environments like a spheroid, for example, which often don't have like a vascular connection within them. And these systems also introduce mechanical and physiological forces such as shear stress, that was a mouthful, shear stress, which is a sort of friction-like force when fluid flows past a surface. And it includes stretching as well, which, for example, you would see in lung expansion during breathing or rhythmic movement in the gut. So already these aspects are incorporated to better mimic what's happening in the human body.
And is there evidence now that this is working? This is kind of a much more functional approach?
Yes, there's quite a few caveats to that. I'll get into those. But right now, these microphysiological systems are being widely used in early drug development as a screening process to say, what is looking hopeful, what's looking more toxic, which candidates do we want to develop farther and, invest the money in.
Right.
So that, farther down the line, we have a higher chance of success. But what's really interesting is that these systems have been created for almost every organ and tissue in the human body, which is
really
honestly incredible. And in some of these instances, we're seeing them actually linked together to form multi-organ connections to get more of a full body picture.
Oh, interesting. Wow.
So, for example, some compounds only present as toxic when the liver and the kidney interact. So if you're just testing on a liver chip or you're just testing on a kidney chip, you might get good data and you might advance that candidate just for it to fail farther down the line. So if you see this interaction earlier on in the process, that's going to give you a lot more insight. That's going to save you time and money.
Very interesting.
Yeah.
Wow.
Yeah. There's a lot of potential here.
Yeah.
Yeah. And they're very, very promising. But sort of like we said, this important caveat is that it doesn't mean they can recapitulate all the important aspects of organs or a full body experiment. At the end of the day, they're still simplified versions of the complexity found in vivo, meaning inside of a living organism.
So how much can this speed up the process of drug development?
Yeah, quite a lot. I mean, if you're getting answers earlier on in the process, that's automatically going to cut that time down a lot. And in terms of the financial component, if you are failing faster, that is going to save you money in the long term. Even if these devices sort of have that upfront cost, it doesn't even compare to what the money that you are going to shell out once you're in, phase 1 or even phase 2 and so on of human trials.
Because, failing in this industry is not it's kind of a given.
Yeah.
It's
Yeah.
like there's more fail than
Yes.
success.
Yeah.
So if you
more
can
than
fail
90%
faster,
failure rate.
yeah, if you can get past that fail faster, that makes sense. You're saving money, you're saving time, and you'll actually, it gives an opportunity to actually find the actual solution in a more realistic time frame.
Yeah,
Interesting.
absolutely.
Wow.
And I will say that another important note is that not all questions need a microphysiological system to be answered. Yes, they are more biologically complex, but in some instances that doesn't necessarily make them better depending on the question that you're trying to answer. So it's all on a fit for purpose basis, right? Because you don't want to use something that you actually don't need.
Right. So now kind of fit Speccro in here. How do you guys fit into this world, and where's your focus?
Yeah. So we use NAMs to solve problems. We help companies incorporate them into their drug development process to answer problems and answer questions in a faster, more time effective, cheaper way. So we help companies figure out what NAM to use, when to use it, which, like I said, will depend on the question and the therapeutic area. And
Right.
we will work on programs that are NAMs light, NAMs heavy, somewhere in between. And the long-term ambition here is to use NAMs to replace existing standards. But having said that, right now there is an important pragmatism. I'll say using them in a complementary fashion with animal models and animal studies, this is kind of our sweet spot right now in drug development.
So you're using them, but you're using them to, you're reducing animal studies, but you're also complementing them.
Exactly. And, also combining different NAMs models to address the shortcomings of each other can have this amazing synergistic effect.
Is there a world where there is no animal testing at some
Hopefully.
point? It could get you. Is that the goal, to get it to that stage?
I think right now that is a little bit of a pipe dream, but never say never. I think we might be a couple decades away from that, even with how fast technology is progressing, because, these systems, like I said, they are more of a piece of the whole puzzle. And even though you're seeing combinations of connections that some people are calling human on a chip, it's still not representative of what's actually happening inside the human body fully. But yeah, I mean, anything is truly possible, and that's the hope, that's the dream. And the government just put out an initiative basically saying in 10 years we want to see no animal testing. I think a lot of scientists, that gives them a little chuckle, because they're like, well, there's a lot of process and a lot of work.
Something's got to happen
that.
between now and then. Yeah,
Exactly.
I mean, we're talking about the animal testing component like it's a very primary thing, but I mean the primary goal is to speed up drug discovery.
Exactly.
Right. The reducing the use of animals in studies or eliminating animals in studies is a really great byproduct, but the primary focus is to get drugs through the discovery stage process faster, cheaper, and be more effective and efficient, so that we can get drugs into the marketplace that are actually going to help patients in
Exactly.
the end. I mean, that's the primary goal, right? That's the animal testing is an awesome idea, but it's also a byproduct of where we're actually trying to get to.
Exactly, yeah. I mean, I don't think the main focus here is the whole ethical component of it,
No.
that's what initially got me in the door. Like I said, I love animals, and
I mean, there'll be a lot of people that will really, really connect with that.
yeah, yeah. And so, I mean, it's like I said, it's there's a lot of positives. There's a lot of benefits that come from this. But, a major limitation right now is that these devices, some of these technologies, methods, processes are not fully accepted by regulators. And
Right.
this is a very fluctuating area. There's a lot of different sectors of drug development right now putting their heads together to sort of redefine this space and what it means, and also just get a handle on the global terminology of some of these things.
Right. And so Speccro is already working with businesses and drug developers in this space, right?
Yes. And we're a startup, we're new. We're about 9 or 10 months young. So we're very
Oh, really? That's pretty young.
Yeah, we're very early on in this process, but we're seeing a lot of momentum, a lot of things pushing us forward and sort of rowing the boat forward.
Are you still in a development phase yourself to a certain extent, or because you're actually working with clients now who are utilizing your services and able to almost validate what you're offering, but you're also still in business growth development yourself, right?
Yeah, definitely. I'm the scientific operations coordinator at Speccro, which is sort of just a fancy way of saying that I'll do a little bit of everything, as is the nature with a startup.
Well, that's what a startup is.
Exactly.
Yeah. Everyone wears many hats.
Exactly.
I think Sashi and I talked about that a lot. He's worn a lot of hats.
Oh, absolutely.
Every day, wears lots of hats, every day almost. Right?
Yes, he is a jack of every trade possible. And not just a jack, like he is a master, truly. But yeah, it's been very interesting. And the learning curve has been steep. But that's how I want it at this phase of life, right? The steeper the learning curve in your 20s, sort of predicts your success in your 30s and 40s.
Well, I mean, look what you did in that 9 months. You kind of found a place to call home in this industry, and you're learning a lot. Plus, you're holding a leadership role with a startup, and you're a woman. So I mean, that's excellent too, right? Yeah, a women leadership, which is also in a predominantly male environment, right?
Yeah, I mean, most of the rooms I walk into are 95% male, which, hasn't really, I'm going to sort of rephrase that, which doesn't faze me anymore, but I had sort of a come to Jesus moment when I went to this event in San Francisco where I met you, actually, at the JP Morgan conference. And it was a women's health focused sort of after party event, sponsored and hosted by Portfolio, who invest in women's health companies. And, I walked into the room and there were about 300 women there, and add some men for some change as well. But it was sort of one of those moments where I just thought, wow, like this is the future. It was just an entire room of women supporting each other and, lifting each other up, being interested and invested in the state of women's health and women's futures in this industry. And it was honestly such a profound experience, and it really made me want to seek those experiences out more.
Well, I think that's, our CEO, Meghna, is, that's why she became a founding member of BLPN, is because it's an environment as well that supports those initiatives and that. And even herself, creating her own sort of women leadership organization called Club Mama Bee. I mean, these are initiatives that are really important and are doing a lot for women leadership and women in business and, women in executive decision-making roles, it's incredible and great. You're doing, it's awesome. I think it's the more the merrier as far as I'm concerned.
Absolutely. Absolutely.
So let's talk about your role at Speccro. You're the what's your title
I'm
again?
the Scientific Operations Coordinator.
Yes. I knew I was going to say it right. I was almost a chief.
Yeah.
But
not yet. Maybe one day.
Yeah, right. You'll get there.
Yeah.
Science Coordination Officer. So that's tell me what your role includes every day.
Yeah. So this role, we sort of had to build from the ground up. I basically created a role for myself, and it has fluctuated and evolved as the needs of the company have changed over time. So it really does look different on a day-to-day basis and week-to-week basis. Some days it's business development, could be marketing, could be admin. I'm trying to dip my finger in a lot of different pots, and one of the initiatives that I'm trying to lead for the company is doing these interview series where I interview different experts in the NAMS industry or NAMS field and really talk about, A, what their specific technology is, B, what they're working on, what the limitations are, what the benefits are, the different nuances, how that's going to look for the next 6 months to a year, maybe even 5 years from now. And even just doing that and being exposed to a lot of different people and their insights and their experience has been a learning opportunity in and of itself for me.
When you say NAMS, do you still get people? Are you getting people going? I don't even know what you're talking about.
Yeah.
Is it that still that sort of new and fresh?
It depends on. I would say, it depends on who. It depends on the event that I'm at. A lot of these events I'm going to are very NAMS geared and will often sort of have that in the title. And for example, there is a conference next month called the MPS World Summit. This is the Microphysiological Systems World Summit, those, engineered devices that I talked about. And, that falls under the category of NAMS, and there's a whole conference about them. Right. So, this is...
It's becoming more commonplace.
Exactly. Okay. And, I think people are starting to recognize that term more and more and sort of realize, what falls under that.
Right. And I mean, that's just momentum. Right?
Exactly.
That's what drives that and expands, and all those areas that you talked about earlier on. They'll just continue to expand and evolve and become more effective.
Mm-hmm. Yeah, absolutely. I'm excited to see what the future has in store. I mean, things are moving fast.
Anything super cool that you see coming that you're excited about, like other things that are on the horizon?
Yeah, I mean, I would say that one of these devices that piques my interest very much is the brain on chip devices. And
okay, let's talk about that. What is that?
Yeah, I mean, the brain is notoriously very complex and hard to study. And so being able to isolate some of these components of the brain and be able to study them more accurately, I think has immense potential. Growing neurons on a chip and testing them for, different CNS diseases or issues that you're going to see is really fascinating.
That is very interesting. And so that's something that you can look forward to work on as well, right?
Yeah. Hopefully. I mean, coming from a neuroscience background, that was a really amazing foundation for me for this entire field, but I always am inherently more drawn to, that sort of niche.
The neuro stuff. Yeah. It's pretty fascinating. And what's next for Speccro? Like, where are you focused right now with Speccro?
Yeah, I mean, getting clients in the door.
Right. Right.
You know,
And so
as always.
when is a client, when's the best time? Where are they at in their process where they go, we need Speccro?
Yeah, I mean, like I mentioned before, this can really range depending on the question they're trying to answer. It can be early discovery, or it can be farther down the line, right before IND. So it really depends on the context.
Right.
Sorry, I know that's a vague answer.
Well, you were very specific earlier on. There are a lot of layers to this.
Yeah.
So, I mean, they will know when they're ready to call you, but they can find you on your website, which is speccro.com, right? SPECCRO.com, and they can always reach out to you on LinkedIn,
Absolutely.
right? Okay. Did we cover everything you wanted to talk about today?
I think we did. Yeah. I guess just to tie it up all in a bow, is, these models have incredible potential to better mimic human physiology. And, there are those limitations that I mentioned, but the opportunities are really endless.
That's cool. Well, thank you so much for sharing all this with me.
Thank you for having me.
You've done a great job sort of explaining a very complex, scientifically complex system, and to me really well. Like, I really felt like I have a much more solid understanding of NAMS,
Awesome.
particularly what the acronym means, the new Approach Methodologies, which
Yes,
is
sir.
very cool. I look forward to catching up with you again, probably at San Diego in Bio San Diego.
Definitely.
Are you going to that?
Yeah, Shashi is. I'm still TBD, but we'll see.
Oh, okay. Yeah. Okay. Well, that's great. Thanks for spending your time with me, and I look forward to talking with you again soon.
Thanks
And
so much
all
for
your
having me.
contact information will be in our show notes, so people can reach out to you if they want to
Awesome.
get more details and more information.
Thank you so much, Heath.